Hey guys, hope you're having a good day today. Don't forget that you need to take your Chapter 14 quiz today. Um, and then you're going to be watching this video. We're going to start Chapter 15 today. Tomorrow and Friday, you'll be working on lab exercise 22. Monday and Tuesday next week, we'll be finishing up Chapter 15. And then you'll be doing your last lab exercise, as well as having your last lab exam. So we're moving right along. This is our last chapter, chapter 15. You are going to be we are going to be talking about the 5 special senses. Remember that our special senses are located in special sense organs, and they include smell, taste, sight, and then hearing and balance. Of course, smell is your nose. That's the special sense organ. Taste is the tongue. Sight is your eyes, and then hearing and balance are both in your ears. And again, remember these are in contrast to your general senses, which is basically your feeling. So that is pressure, touch, pain, proprioception, heat, cold, things like that. Okay, so our sense of smell is called olfaction, and basically it occurs in response to odors that are going to be stimulating sensory receptors in the olfactory region of your brain. The olfactory region of your brain is in the frontal lobe. It's basically that olfactory lobe that is on the inferior portion of your frontal lobe. In the extreme superior region of your nasal cavity, you have a tissue called olfactory epithelium. In that olfactory epithelium, you have about 10 million primary olfactory neurons. The ends of these neurons, or the ends of these dendrites, have enlarged ends called olfactory vesicles. And these olfactory vesicles have little cilia on them called olfactory hairs. And those hairs are embedded in mucus. And so what's going to happen is that the odorant molecules from a substance, will enter into your nose. They will go up there to the extreme superior region of your nasal cavity, and they will dissolve in the mucus that covers this olfactory epithelium. And once they've dissolved in that mucus, they will attach to receptors on those olfactory hairs, which are on those olfactory vesicles, and then this will stimulate those olfactory neurons, those primary olfactory neurons. It will generate an action potential, which will then be carried up to by cranial nerve 1, the olfactory nerve, up to the olfactory lobe on the inferior side of the frontal lobe of your cerebrum. So here's a picture showing us, again, here's our nose, and in the extreme superior portion here, we have this olfactory epithelium. Again, this olfactory epithelium has about 10 million of these primary olfactory neurons, so it has a lot of these neurons. But anyway, here are our olfactory neurons. We can see it here. We have here here's the actual olfactory neurons here in this olfactory epithelium. Here are the olfactory vesicles, and then this is an olfactory vesicle, and then there's little cilia on the end there. So the receptors for the odorant molecules are here. There's a clear layer here. You can see that is going to be the mucus. The olfactory vesicles contain those cilia that are embedded in that mucus. The odorant molecules will dissolve in this mucus, bind to these olfactory hairs on this olfactory neuron. This will end up generating an action potential in this olfactory neuron, which will then, that action potential will be carried up these olfactory nerve processes to the olfactory bulb, which is on the inferior portion of the cerebrum. So again, as I just said, the odorant molecules of the chemical that you're smelling dissolve in the mucus. They attach to the olfactory hair membrane receptors, which are chemoreceptors. Again, they bind chemicals, and these receptors are on those olfactory hairs, which are the cilia on those olfactory vesicles. And then these receptors are associated with something called a G protein mechanism. Basically, there's a G protein that will activate something called adenylate cyclase. This will form CAMP, And then this CAMP will go to sodium and calcium channels and open them. When those channels are open, sodium and calcium are able to move into those olfactory hairs, those cilia, and this generates an action potential in that olfactory neuron. We have lots of different receptor molecules for these different odorants that can be in chemicals that we smell. There's about 1,000 different odorant receptor molecules. So, and then coupled with the fact that there's multiple G protein mechanisms, multiple G protein pathways, we have at least 4,000 detectable smells. Probably more than that, but at least 4,000. Another thing, our sense of smell is very sensitive because it takes very few odorant molecules binding to those olfactory hairs in order to cause this whole chain reaction to happen and trigger an action potential. Something that we talked about yesterday when we talked about this adaptation. An example of adaptation is olfactory fatigue, or we call that being nose blind. Basically, how this happens, how this adaptation occurs, is that those odorant receptors on those olfactory hairs, those cilia, will become saturated with odorant molecules. This means that they're all the receptor molecules are full of odorant molecules. And once all of those receptor molecules on those cilia become full of odorant molecules, then they're not going to generate action potentials anymore. And so this causes you to become this causes that adaptation. It causes you to become less sensitive to the odor the longer you're exposed to it. And so we call that being nose blind. You've kind of gotten used to that smell because all of your receptor molecules for that odorant on those olfactory hairs are full of that odorant, and so they quit generating action potentials and you don't notice that smell anymore. Okay, this is showing you the G protein mechanism. Here we have an odorant molecule binding to a receptor on those olfactory hairs, those cilia, the membrane of those cilia. What this does is this activates this G protein mechanism. This alpha subunit of this G protein mechanism will go over here to this adenylate cyclase, as you can see here, and it activates it and forms CAMP. It uses ATP to form CAMP, and then the CAMP goes over here. This is a sodium ion channel, but it's not showing a calcium ion channel, but it goes over here to the sodium and calcium ion channels and triggers them to open. And then when those sodium and calcium ion channels open, sodium and calcium come into the cell, it depolarizes the cell, and an action potential is generated. Again, we talked about this in regular anatomy last year. Remember that your olfactory epithelium is directly connected to your brain. So that is your nose, the extreme superior portion of your nose, that olfactory epithelium is the only place where your brain is directly exposed to the environment. And so therefore, there's a lot of wearing down and being replaced of these neurons in that olfactory epithelium. In fact, the entire olfactory epithelium completely breaks down and is replaced entirely about every 2 months. And the cells of that olfactory epithelium, the basal cells of that olfactory epithelium are going to replace the olfactory neurons in that olfactory epithelium. And remember that most neurons are permanent cells, meaning that once they mature, they never divide again their entire lives. But these olfactory neurons do. So this makes them very unique because most neurons do not repair, they do not replace themselves. They don't have the ability to do that. But these olfactory neurons in this olfactory epithelium can. Okay, so the specific pathway that the action potential takes to get to the brain, you start again with those that odorant molecule binding to the receptors on those olfactory cilia, those olfactory hairs, and then this generates an action potential in the primary olfactory neurons in that olfactory epithelium. And again, these are those rare bipolar neurons. You have them in your nose, and you also have some in the retina of your eye. We'll talk more about that next week. So the primary olfactory neurons, which are bipolar neurons, that's where the action potential is generated first. And then from there, that action potential is carried along cranial nerve 1, which is your olfactory nerve, to your olfactory bulb on the inferior portion of your frontal lobe. In the olfactory bulb, secondary, the action potential goes to secondary olfactory neurons, and then through olfactory tracts to that the olfactory cortex, which is in the frontal lobe of your brain. Again, remember that smell is the only special sense that we have that does not go through the thalamus. So again, it goes directly from the primary olfactory neurons in that olfactory epithelium up the olfactory nerve to the olfactory bulb, along secondary olfactory neurons and olfactory tracts to the olfactory cortex of the frontal lobe, directly to that frontal lobe to be interpreted, and never goes through the thalamus first. Other places that action potential for that odorant, for that smell, is going to go besides your olfactory cortex, it's going to go to the amygdala, it's going to go to the hippocampus, the limbic system, the hypothalamus, which are secondary olfactory areas. And basically, again, remember that the amygdala is associated with emotion. So this is, how smells can trigger emotions. It goes to the hippocampus. This is how smells trigger memories. So when I smell Scotch tape at Christmas time, then it brings back those good emotions, because the smell of that Scotch tape, those action potentials aren't just going to the olfactory cortex so that I interpret that smell and I think, oh, hey, this is Scotch tape that I'm smelling. But those action potentials also go to the amygdala to trigger that nostalgia, that good feeling of, oh, remember, that's that good warm feeling that I get when I smell that. And then those action potentials from that smell also go to my hippocampus, triggering the memories of Christmas when I was at home, when I was a child with my brother and sister and my parents. So I get those. This is how smells trigger emotions and memories, because the action potentials are not only going to the olfactory cortex in the frontal lobe, but they're also going to these other parts of the brain that are stimulating emotions and memories as well. And so this is how odors and smells are able to evoke memories and emotions from you. Again, this is just a visual showing you how this happens. Again, here are those, the olfactory neurons will be right here in this olfactory epithelium. Then we will go to the cranial nerve 1, and then we will go to the olfactory bulb, secondary olfactory neurons, then the olfactory tract up here into the olfactory cortex, which is in the frontal lobe. Again, this is my olfactory bulb. This is my olfactory bulb on the inferior portion of the frontal lobe. Here's the frontal lobe. Here's my olfactory tracts. And then here is my olfactory cortex in the frontal lobe as well. So again, the thalamus is on back here. Smells never go through the thalamus. Again, pretty much we have the brain directly exposed to the environment right here in the extreme superior portion of your nasal cavity. Okay, so let's talk about taste now. Taste is called gustation. We have on our tongues, we have taste buds. Taste buds are the sensory structures that contain the gustatory cells that detect taste stimuli. So the receptor cells that receive taste stimuli are gustatory cells, and they are inside of a taste bud. Okay, these taste cells these gustatory cells that are inside this taste bud have little cilia. Well, they're not cilia. These are not cilia. The olfactory hairs are cilia. These are not cilia. These are microvilli. Remember, cilia can move. Okay, microvilli don't move. They look a lot like cilia, but they don't move. We talked about these as being cell extensions back at the very beginning of the class. We call these little microvilli that are on these gustatory cells, we call them gustatory hairs. And these extend into something called a taste pore. And the taste pore is kind of the opening of the taste bud to the environment. So basically these molecules that we're going to be tasting, they're called tastants. They're going to enter that taste pore and they're going to bind to receptors on these gustatory hairs, and then we're going to end up generating an action potential. Again, these taste cells are not neurons. Okay, they are cells that have receptors. So these are going to be these are not going to be. Yesterday we talked about those primary and secondary. What was it that we called it? I don't remember. But anyway, one of them involved actual releasing of neurotransmitters, and then the other one involved directly carrying the information. So like with our smell, it's the one, and I don't remember whether it was primary or secondary. You'll have to go back and look at that. But anyway, it's the one where you don't release neurotransmitters to generate an action potential. The action potential is generated by that G protein mechanism when the molecules bind to those olfactory hairs of the olfactory neuron. Okay, so we don't release transmitters there. The action potential is generated by the odorant binding to the receptors on that olfactory hair. Okay, our taste is going to be different, all right, because these gustatory cells are not neurons. So we don't generate the action potential in the gustatory cell. The gustatory cell will release neurotransmitters to generate an action potential. And I think that was the secondary. I think it was. So, but you'll have to go back and look. I may have it backwards. But anyway, I think the smell is more primary, because when the odorant molecules bind to that olfactory neuron, we get the action potential and it gets generated. Okay, we're not generating an action potential in the gustatory cells. We are going to cause those gustatory cells to release a neurotransmitter that will then generate an action potential in a cranial nerve. Okay, so anyway, the bumps that we have on our tongue, guys, are not taste buds. They are called papilla. Okay, the taste buds are going to be smaller than those papilla, and they're located at the edges of those papilla. So the bumps on your tongue are not your actual taste buds. The bumps that you see are papilla, and the taste buds are much smaller. You can't see those, and they're on the edges of those bumps on your tongue. You also have taste buds, which I didn't know until I started teaching this class. You have taste buds on your hard and soft palate, which is the roof of your mouth. You have taste buds on your lips and your throat. And so, you think, well, I'm going to try to swallow this without getting it on my tongue because I don't want to taste it. You're still going to taste it, because if you get it on your throat, you have taste buds on your throat. So it's crazy. And I'm thinking that children have more taste buds on other places than just their tongue. The older you get, the more you have them mainly on your tongue and not as much on your lips and throat and things like that. I think children have them more everywhere. Okay, so the papilla. Again, the bumps on our tongue, we have different kinds of bumps on our tongue based upon and we're going to be classifying them according to their shape. The first kind are called filiform. The filiform are more filament shaped. They are the most numerous papilla, the most numerous bumps on our tongue, and there are no taste buds associated with those. So why in the world do we have those filiform papilla? Well, the reason we have them is that they just give the tongue a texture. They give it a rough surface so that where food is able to cling to it better, and and your tongue can participate in manipulating the food and forming that bolus that you're going to swallow. Valate papilla are the largest papilla on our tongues. There's not very many of them, about 8 to 12, and they're on the posterior part of your tongue mainly, and they do have taste buds. The foliate papilla are named foliate because they are leaf-shaped. Like the foliage is the greenery on a tree, so they're leaf-shaped. And they are in the folds on the side of your tongue, and they contain the most sensitive taste buds. And these are the ones that decrease in number with age. So as we get older, our taste overall, our sense of taste becomes less sensitive because we do lose taste buds as we get older. And these are the ones that we're going to be losing. The last type of papilla are fungiform papilla. They are mushroom-shaped, and they are irregularly scattered over the superior surface of your tongue. They look like small red dots, and they also have taste buds. So the only type of papilla that don't have taste buds are the filiform papilla. Those bumps are mainly there just to help you manipulate your food with your tongue. They help the food cling to the tongue better. The others do have taste buds, with the foliate having the most sensitive taste buds and being the ones that decrease with age. So here we see in our picture the valate, again, the largest, are back here on the posterior portion of your tongue. Then we have the foliate, the ones that are kind of leaf-shaped, are here on the lateral side, the lateral portions of the tongue. Then we have the fungiform, which are the mushroom-shaped, and they're more here on the tip of the tongue. And then we have the filiform, which are more filament-shaped, and they're kind of scattered irregularly around the tongue. Or no, those are going to be the fungiform that are scattered irregularly. Maybe the filiform are the ones that are on the tip. Okay, so again, our taste buds are on the edges of those papilla. They are oval and they are embedded in the epithelium of the tongue and the mouth, and you have about 10,000 taste buds in your oral cavity. So again, remember you have about 10 million olfactory neurons in that olfactory epithelium. We have only about 10,000 taste buds in our oral cavity. So again, our taste is not nearly as sensitive as our smell is. We have three major types of specialized cells in a taste bud. The major type that we're going to be worried about are those gustatory cells. As we mentioned earlier, you have about 50 gustatory cells in a taste bud, and these are the actual sensory cells that detect taste. The other two types of cells, the basal cells and supporting cells, are not sensory cells. So it's the 50 gustatory cells that are the sensory cells that detect taste. And again, they do have those microvilli, which we call taste hairs, that extend through an opening in the epithelium on the tongue and the mouth, and this little opening is called the taste pore. So these taste cells, like those olfactory hairs of those olfactory neurons, these taste cells of these gustatory cells, these taste hairs of these gustatory cells are directly exposed to the environment, and so they wear down quite frequently and are replaced continuously. However, where we said the olfactory epithelium is completely replaced about every 2 months, these olfactory cells are going to be, their lifespan, the lifespan of these taste cells is probably a little bit shorter because they are going to be going through more friction and wear and tear because of the food in your mouth rubbing on them. So their lifespan is about 10 days as opposed to, probably longer for those olfactory cells. Okay, so here we can see this opening is the taste pore. Here we can see extending through the taste pore is the taste hairs. These little taste hairs are going to be, this is the actual gustatory cell right here, the pink. And then at the end, you can't really see it here, but you can see it here. You can see those taste hairs, which are the little microvilli extending through the taste pore. So what's going to happen? A molecule from the food that you're eating called a tastant is going to dissolve in your saliva. Again, that's one purpose of saliva. It dissolves the molecules in the food that you're eating. So those tastants dissolve in the saliva. They go through that opening, this taste pore, and they bind to receptors on these little taste hairs and stimulate these gustatory cells. And notice these little gustatory cells. They have synaptic vesicles containing neurotransmitter. And so that neurotransmitter will then go and stimulate this sensory neuron here. So the neurotransmitters are released by the taste cells. This stimulates action potentials in the axons of secondary neurons associated with them. So this is a secondary neuron associated with this gustatory cell. So when the tastant binds to receptors here on the taste hair, this is not going to generate an action potential like it does in those olfactory hairs, those olfactory neurons. What this does is it stimulates this cell to release neurotransmitters. Those neurotransmitters will bind to these secondary neurons here, and that will generate an action potential in this secondary neuron. So let's talk about the taste. Again, remember, we only have about 10,000 taste buds on our tongue and in our mouth as opposed to 10 million olfactory neurons in that olfactory epithelium. So therefore, our sense of smell is much more sensitive to a variety of different odors as compared to our sense of taste. Our sense of taste basically can only detect 5 different tastes, and those are sour, salty, sweet, bitter, and umami, which is a savory taste. The sour receptors are the most sensitive receptors, and they are on the lateral aspects of the tongue. Basically what this means is that the lateral sides of your tongue are most sensitive to sour. Okay, what we're going to see is that all of these taste buds are sensitive to all 5 tastes, but some are more sensitive to a certain taste. They can taste all tastes, but they're more sensitive to certain tastes. So basically, the taste buds on the lateral portions of your tongue are going to be most sensitive to sour. Now we're going to see that these taste receptors are sensitive by different mechanisms, okay? We're going to see that these last 3 use a G protein mechanism similar to what is used by those olfactory neurons as compared to sour and salty. Sour and salty are going to be, they're going to involve ion channels opening up to generate that action potential. So basically, your taste buds on the lateral aspects of your tongue are most sensitive to sour. Basically, remember, acids taste sour. Well, why? Hydrogen ions in acids end up causing depolarization of that taste cell and causing the release of that neurotransmitter to generate that action potential. How do the hydrogen ions do this? Well, number one, they can enter the cell. They can enter that taste cell, that gustatory cell, through hydrogen ion channels. Another thing they can do, they can bind to potassium ion channels and prevent potassium from exiting the cell, and that causes depolarization of the gustatory cell. They can also bind to and open other ion channels, allowing those ions to enter the cell and generate an action, not generate an action potential, but stimulate that cell to release its neurotransmitters so that an action potential can be generated. Okay, salty. The most sensitive taste buds for salty are on the tip of your tongue. This salty and sweet are the two tastes that we have the least sensitivity to. Again, salty is going to be similar to sour, except that it's not hydrogen ions that end up depolarizing the gustatory cell and causing that neurotransmitter to be released, but it's sodium ions. Sodium ions or other metal ions end up depolarizing that gustatory cell and causing it to release that neurotransmitter. Salty is craved by humans due to our need for minerals, due to our need for metal ions. You know, we need metal ions like magnesium, we need zinc, we need sodium, we need calcium. So all those metal ions in foods are going to cause the salty taste. And again, the salty is craved by humans because, and that it is believed, like an evolutionary theory, it is believed that is due to our need for these minerals. So we crave foods that have this salty taste because when we eat this salty taste, we're getting these metal ions that our body needs to function. Okay, our next taste is bitter. And again, these last three are going to be using not ions, but the G protein mechanism like we discussed with the olfactory sensation. So the most sensitive receptors for bitter are on the posterior aspect of your tongue. Again, that's going to be toward the back. And again, these the gustatory cells are stimulated by bitter to taste bitter through a G protein mechanism. Okay, remember that's where the alpha subunit activates adenylate cyclase, forming C-AMP, which then is going to open up channels to cause depolarization. We have the highest sensitivity to bitter, and this is thought to be, again, an evolutionary adaptive mechanism because many things that taste bitter are toxic. They're poison. So we don't crave bitter. Bitter tastes bad to us. We don't crave it, and in fact, we don't like it. And this is thought to have an evolutionary adaptive mechanism as far as because a lot of things that taste bitter are poison to us. So it actually protects us. That bitter taste protects us from eating things that would hurt us, or protects animals from eating things that would hurt them. And again, most things that taste bitter have alkaloids in them, and these alkaloids stimulate this G protein mechanism to cause that gustatory cell to release neurotransmitters. Okay, the next one is sweet. The tip of the tongue has the receptors that are most sensitive to sweet. Again, this also ends up resulting in depolarization of the cell through a G protein mechanism. Again, along with salty, we have our lowest sensitivity to sweet and salty. Sugars, carbohydrates, and some proteins actually give us this sweet taste. And again, this is another taste that is craved by humans due to our need for sugars to give us energy. So again, evolutionarily speaking, it is thought that we crave that sugary taste because that helps us to take in, or we crave that sweet taste because that helps us to take in sugars that we use for energy. The last taste is that umami, that savory. Amino acids such as glutamate are going to end up causing depolarization of the gustatory cell by a G protein mechanism for us to be able to taste umami. And again, this is another taste that is craved by humans due to our need for protein. So evolutionarily speaking, it is believed that we crave the savory taste and so that way we will eat things that taste savory, and that savory taste is caused by amino acids, which are part of proteins, and so that ensures that we get proteins in our diet because our body needs amino acids, our body needs proteins. So again, for 4 of these 5 tastes, it is believed there's an evolutionary mechanism that either causes us to crave that taste or causes us to not like that taste. So, for example, again, humans crave salty because things that taste salty have these metal ions in them, which are things that we need in our diet to survive. Humans crave this sweet taste And that's what gets us to eat things that taste sweet so that we have sugar in our diet to give us energy. Humans crave this savory taste, and it is amino acids and proteins that give us that savory taste. And in this way, it ensures that we take in proteins that we need in our diet. Humans do not crave, but actually dislike this bitter taste. And that helps to make sure that we don't eat things that are bitter, again, as a protective function, because many things that are bitter are poison to us, and we don't need them in our diet. Okay, so again, how do these things work? If we're looking at ions such as the metal ions with our salty taste, or the hydrogen ions with our sour taste, basically those ions are going to basically open up channels and go through ion channels and cause depolarization of that gustatory cell, which then releases neurotransmitters and generates an action potential in that secondary neuron. Taste, such as your sweet, your bitter, and your umami, when that tastant binds to a receptor in that gustatory cell on the gustatory hairs, or those taste hairs, that molecule binds on that receptor, then this activates the G protein. The G protein activates the adenylate cyclase to produce C-AMP, which then is going to open up an ion channel to let ions in, again, to depolarize that gustatory cell so that it will release its neurotransmitter and generate an action potential in that secondary neuron. There are things that affect taste. One thing that affects taste is texture. Our perception of taste is texture. Another is temperature. I know, for example, even I think about sweet tea. Sweet tea tastes different when it's hot than it does when it's cold. There's some things that we like, we would rather have cold because they taste better. Other things we'd rather have warm because they taste better. Again, so temperature affects our perception of taste. Also texture. I am a big texture person. This is why I really don't like ground beef much at all, because I'm big on texture. So texture affects the perception of taste, and that can cause you to like or dislike certain things. Also, taste has a very rapid adaptation. So for example, I noticed this myself. When I drink a cup of coffee, I like things really sweet, but I noticed that, after I've taken a few drinks of it, it doesn't taste as sweet to me anymore. That's because my taste buds, my gustatory cells are becoming adapted to that sweet taste. Again, all taste buds are sensitive to all 5 tastes. Each taste cell is within the bud. Okay, so in the bud you have about 50 gustatory cells. So each one of those cells is going to be sensitive to one of the 5 tastes. So a taste bud can taste all 5 tastes because of those 50 gustatory cells inside that taste bud. Some of those gustatory cells in that taste bud are sensitive to sweet, some to sour, some to bitter, some to, so you have each gustatory cell is going to be sensitive to a specific taste. But since you have at least 50 gustatory cells in a taste bud, that you have different gustatory cells that are sensitive to different tastes in that taste bud. So taste buds can taste all 5 tastes. And again, remember that our senses of smell and taste work together. So a lot of what we taste when we eat something is due to the smell that we're smelling while we're eating it. Okay? Remember that our sense of smell is much more sensitive than our sense of taste. So our sense of smell contributes to our sense of taste because we can only taste 5 flavors. Also, different tastes have different thresholds. Again, meaning that we're more sensitive to some tastes than others. We are least sensitive to sweet and salty. We are most sensitive to bitter. And again, this is thought to be an adaptive mechanism. It ensures that we don't take in things that taste bitter because we don't want to take, because a lot of things that are bitter are poison, and it keeps us from eating something that would harm us. Okay, so what are our pathways that taste takes to get to our brain? Well, basically, there are 3 cranial nerves that are involved in tasting. The one part of our cranial nerve 7, the facial nerve, carries sensations from the anterior 2/3 of our tongue, so that's most of our tongue from the front back. And then our glossopharyngeal nerve carries taste to our brain from the other 1/3 of our tongue in the back, the posterior 1/3. And also our pharynx, which is our throat. And then the vagus nerve, cranial nerve 10, carries a few fibers for taste from our epiglottis, which remembers that little flap that folds back to cover your trachea as you swallow. So again, you even have some taste cells, some taste buds on your epiglottis and your throat, your pharynx. So basically, from your gustatory cell, those neurotransmitters will go to the secondary neuron that is going to be in these cranial nerves. So from taste buds to the cranial nerves, then that's going to go to the medulla oblongata, the tractus soleratus of the medulla oblongata, and then to the thalamus. Remember, all sensations except for smell go through the thalamus. And then from the thalamus, it goes to the insula of the cerebrum. Remember, the insula is the fifth lobe of the cerebrum. We said mainly the temporal lobe, the insula of the temporal lobe was responsible for tasting when we talked about the cerebrum, but this insula is actually kind of in between our temporal and parietal lobes. So if we look at it here, we see that here we have our taste buds are going to be here on our tongue. Again, here is our cranial nerve 10. That is, that's 5. That is innervating our anterior 2/3. Then here is our cranial nerve 9 that is innervating our posterior 1/3 as well as our pharynx. And then here is our cranial nerve 10, that vagus nerve that is also doing the pharynx as well as the epiglottis. So the action potentials will be the gustatory cells and the taste buds will be depolarized. They will release neurotransmitters, which will then activate secondary neurons in these cranial nerves, 5, 7, 9, and 10. And then the action potentials from these cranial nerves will go to that tractus solitarius in the medulla oblongata, and then from there, those action potentials will then be sent to the thalamus here, kind of in the center of our brain, and then from the thalamus to the insula, which is this fifth lobe of the cerebrum between this temporal lobe here and the parietal lobe up here. Okay, and that's going to be all that we're going to be talking about for today. Again, don't forget, if you haven't yet, take your Chapter 14 quiz, and then on Monday we will be finishing up, or Monday and Tuesday we will be finishing up with our Chapter 15. You'll also next week be doing lab exercise 22, and then by next Friday we will be having our last lab exam, so we're almost finished. Please let me know if you have any questions or need any help. I'm here for you. Just let me know. Hope you have a great rest of your week, and we'll talk to you later. Bye-bye.